There is this underlying assumption we all kind of carry around about the whole process of getting older.
Right, yeah.
It's an assumption of just pure thermodynamic inevitability. Like,
Yeah.
we tend to think of the human body like I don't know, a classic car left out in a field.
Yeah, or like a complex piece of machinery that's just, slowly and unavoidably breaking down over time.
Exactly. And the logic feels really intuitive, right? Like, you use your joints over decades, the cartilage wears thin,
Right.
you process oxygen to survive, and you accumulate these free radicals that damage your tissues.
Yeah, so the consensus has always sort of been that aging is just this one-way street of wear and tear.
Right. And the absolute best you can hope for is to just, maybe slow down the rusting process a little bit.
And for the longest time, that was the prevailing dogma in biology.
It makes sense, though.
It does. It's comforting in a strange way because it's just so simple to understand. We viewed aging as this accumulation of random, irreversible damage.
Like, you get a dent in the fender, you lose a spark plug.
Yeah, and eventually the engine just stops. And biologists actually spent decades trying to map out exactly how this damage accumulates, largely assuming that, well, once a cell was broken by the passage of time, it was permanently broken.
But then you step into the world of modern biogerontology, and suddenly that whole rusting machine metaphor just completely falls apart.
It really does.
Because when you actually look at the sources we've compiled for this deep dive today, we are looking at a biological landscape that is, quite honestly, shocking.
It's a total paradigm shift.
Yeah. The science is revealing that aging is not just a passive accumulation of physical damage. It is a highly regulated, plastic, and potentially reversible software program.
And the sources really represent that fundamental shift.
Yeah.
We are moving from a medical framework that simply observes and attempts to mitigate the decay of the human body
Right.
to one that is actively learning how to rewrite the code that governs that decay.
Which is wild.
It is. And to be clear, the goal outlined in all this research is not simply extending lifespan, just dragging out the final years of decline.
Yeah, nobody wants that.
Right. The focus is entirely on health span. It's about maximizing the number of years you are actually healthy, functional, disease-free, and biologically resilient.
Which is exactly why we are taking this deep dive into the cutting-edge science of epigenetics and anti-aging today.
Absolutely.
So if you are the kind of listener who loves those sudden aha moments and you want to bypass all the dense academic jargon to understand the real mechanical levers of human longevity,
You're in the exact right place.
you really are. We are going to explore how our biological clocks actually tick at the cellular level, and more importantly, how emerging science ranging from experimental CRISPR gene editing all the way down to just your daily sleep habits.
Yeah, the everyday stuff.
Right. How all of that is proving that the aging process is a modifiable behavior.
But, before we can talk about how to reverse the aging process, we have to establish exactly what is breaking down.
Right. We need to move from the abstract concept of getting old to the physical microscopic reality of what scientists call the epigenome.
Exactly.
So if we imagine that the DNA sequence, the billions of A, C, T, and G nucleotides you inherit from your parents, is the hardware of a computer, completely fixed at conception,
Right.
what exactly is the epigenome?
The DNA is the hardware. The epigenome is basically the software operating system.
Okay, the software.
Yeah. It sits directly on top of your DNA and tells your cells exactly how to use that hardware. Think about this. Every single cell in your body, whether it's a beating heart cell, a neuron firing in your brain, or a skin cell.
They all have the same DNA.
Exactly. They all contain the exact same hardware, the identical DNA sequence.
Hmm.
So if the hardware is identical, why do those cells look and act entirely differently?
Right. A heart cell doesn't look or behave anything like a brain cell.
And that differentiation is entirely due to the epigenome. It's a complex, dynamic layer of chemical modifications that dictates which specific genes are turned on and which are permanently turned off.
In any given cell type.
Right. It's the instruction manual that tells the hardware what programs to run. And it achieves this highly precise regulation through a couple of primary mechanisms that are just crucial to understand if we're going to talk about anti-aging.
Okay, so let's get into the physical mechanics of this. What does a piece of biological software actually look like under a microscope?
Well, the first and arguably most studied mechanism is DNA methylation.
Methylation, okay.
Yeah. Imagine tiny chemical tags specifically methyl groups, which they're just a single carbon atom attached to 3 hydrogen atoms, attaching directly to the DNA molecule itself.
Oh, directly onto it.
Right. They usually attach to cytosine bases that are situated right next to guanine bases.
Mm-hmm.
And these methyl tags, they act very much like localized dimmer switches on your genes.
Wait, how do they do that physically? Like, do they block the gene from being read?
Exactly that. It's a process called steric hindrance. When a gene has a high concentration of these methyl tags clustered around its starting line, what we call its promoter region,
Right.
the sheer physical bulk of those tags prevents the cell's reading machinery from attaching.
Ah, so it's literally just in the way.
Yep. The dimmer switch is turned all the way down, the gene is silenced, it's locked away, and cannot be expressed into a protein.
Okay, so methylation basically equals silencing.
Generally, yes.
So, like, if I am a skin cell, I want the genes that code for liver enzymes to be heavily methylated. I want them permanently turned off.
Correct. You don't want your skin cells trying to act like your liver.
That sounds messy.
Very. Now, the second mechanism involves how the DNA is physically packaged within the cell. Because your DNA is incredibly long.
Right.
If you took the DNA from just one single microscopic human cell and stretched it out end to end, it would be roughly 2 meters long.
2 meters of genetic code stuffed inside a nucleus you can't even see with the naked eye. I mean, that requires some serious biological origami.
It really does. It requires extreme compression. To fit inside the nucleus, the DNA strand is tightly wound around specialized structural proteins called histones.
Histones.
Right. Think of histones like tiny molecular spools. 8 of these histone proteins clump together, and the DNA wraps around them just like thread.
Okay, I can picture that.
So if the DNA is wound very tightly around these spools, the cell's reading machinery cannot access the genes hidden inside those loops.
They're tucked away.
Exactly. They're locked away in a highly condensed structure called heterochromatin.
So the spool itself acts as a security vault.
Yes, but the vault can be opened through chemical modifications to the tails of these histone spools, specifically adding a chemical called an acetyl group. The electrical charge of the spool changes.
Oh, interesting.
Yeah, the spool becomes less positively charged, which causes it to actually repel the negatively charged DNA strands.
So that pushes it away.
Right. The spool physically relaxes, the DNA unwinds a bit, opening up the chromatin, and suddenly those hidden genes are accessible. The reading machinery can dock, and the gene is turned on.
Wow. Okay, so you have chemical dimmer switches directly on the DNA sequence, and you have these physical spools controlling access to entire sections of the genome.
Exactly.
I mean, that is an incredibly elegant, highly controlled system, but kind of returning to our original problem here. What happens to this pristine software operating system as decades pass?
Well, this brings us to a phenomenon known in the literature as epigenetic drift.
Epigenetic drift.
Yeah. Throughout your life, your cells are exposed to environmental stressors, metabolic byproducts, UV radiation, and just the intrinsic chemical noise of cellular division.
Just living life.
Exactly. And over decades, the precise, youthful patterns of those methyl dimmer switches and histone spools begin to degrade. The system loses its fidelity.
So it gets sloppy.
Very sloppy. The tight spools of silenced DNA, that heterochromatin vault we just mentioned, start to slowly loosen up.
Okay, wait. If those spools loosen up over time, that means genes that are supposed to be locked away forever, like the liver enzyme in the skin cell, might suddenly get exposed and accidentally turned on.
That is precisely what happens. And gerontologists refer to it as transcriptional noise.
Transcriptional noise.
Yeah. The skin cell accidentally starts producing traces of proteins it shouldn't be producing. The cell basically loses its crisp, defined identity. It gets biologically confused.
Oh, wow.
And at the exact same time, genes that should be active, like the crucial genes responsible for repairing DNA damage or generating cellular energy,
They get turned off.
right, they might accidentally accumulate methyl tags and get turned down or silenced completely.
The software is glitching.
Exactly.
You know, it reminds me of an analogy. It's like a symphony conductor who has been leading a massive orchestra perfectly for decades.
I like this.
But slowly, year after year, he starts losing individual pages of his sheet music. Like a violin starts playing a measure early, the brass section misses a cue and stays completely silent. And what used to be a coordinated masterpiece just slowly devolves into chaotic, overlapping noise.
That analogy perfectly captures the systemic failure. And this chaotic epigenetic noise has catastrophic downstream effects on the body.
Like what?
Well, this software glitch leads directly to the physical hallmarks of aging that we can see and feel. Because the DNA repair genes are accidentally silenced by stray methyl tags, your cells lose the ability to fix daily damage.
So genomic instability sets in.
Exactly. Actual physical damage to the hardware begins to accumulate faster. And because the metabolic genes are dysregulated, you get mitochondrial dysfunction.
So you have less energy.
Right. Your cells can no longer produce ATP energy efficiently, which leads to systemic fatigue.
And doesn't this chaotic noise eventually trigger the creation of those zombie cells we hear so much about in longevity circles?
Yes, it is the primary driver of cellular senescence.
Senescence, right.
When a cell accumulates too much of this epigenetic noise and the resulting DNA damage reaches a critical threshold, the cell realizes it's dangerously close to becoming cancerous.
Oh, so it hits the brakes.
Exactly. It pulls an emergency brake. It permanently stops dividing. But instead of undergoing apoptosis, which is normal, programmed cellular death, it refuses to die.
Hence the zombie name.
Right. It just sits there in the tissue.
Yeah.
And worse, it begins secreting a highly toxic cocktail of inflammatory molecules, degrading enzymes, and immune modulators.
So it's actively poisoning the tissue around it.
It creates a localized zone of destruction.
Yeah.
As these senescent zombie cells accumulate in your tissues over decades, they create a state of chronic, low-grade systemic inflammation throughout the entire body.
Which researchers call inflammaging, right?
Exactly, inflammaging.
Yeah.
And this chronic inflammaging then bathes neighboring healthy cells in toxic stress, which accelerates their epigenetic drift, creating a vicious compounding cycle of systemic decline.
Man. Okay, but let's pause and look at the massive implication of everything we just discussed.
Sure.
If the hardware, the actual underlying DNA sequence is still largely intact, but the software is just glitching due to misplaced methyl tags and loose spools, doesn't that fundamentally imply the system is fixable? I mean, if I have a buggy app on my phone, I don't throw the phone away.
No, you don't.
I download a patch or I reboot the system. So can we reboot a human cell?
That realization is the engine driving the entire longevity field today.
It's huge.
It is. If aging is fundamentally an epigenetic software problem, then aging is inherently plastic. It is modifiable. It's not a one-way street of irreversible thermodynamic decay.
That's so optimistic.
It's incredible. But before researchers can develop therapies to reboot the system, they face a massive diagnostic hurdle.
Which is?
We need to know exactly how badly the software is malfunctioning at any given moment. We have to be able to quantify the glitch.
Which brings up a fascinating problem. Because if aging is this fundamentally abstract degradation of microscopic chemical tags, how on earth do scientists actually measure it? Like, we all know our chronological age. You look at a calendar, you count the birthdays. But biological age, the actual state of your software, seems so much more elusive. How do you measure the age of an epigenome?
Well, the first step is separating chronological age from biological age.
Okay.
Chronological age is simply a measure of time passing, right? It's the number of times you have orbited the sun.
Simple enough.
But biological age, however, is a measure of the actual physiological wear and tear on your cellular function. It measures how old your cells act.
Okay.
You could be 50 years old chronologically, but if your epigenetic software is highly preserved and functioning perfectly, your biological age might be 40.
Wow, 10 years younger.
Exactly.
Yeah.
Conversely, if you've lived a life of extreme stress and poor diet, your biological age could be 60.
So how do we actually read that biological age from a person? Because I can't just look at someone and accurately guess the state of their cellular spool.
No, you can't. We read it through what the field calls epigenetic clocks. And the evolution of these clocks over the last decade is one of the most rapid and astonishing developments in modern biology.
So how did they start?
The original first-generation clocks act a lot like a car's odometer. Researchers took thousands of tissue samples and used advanced machine learning algorithms to scan hundreds of thousands of specific DNA methylation sites.
Those little chemical dimmer switches?
Exactly.
They just let the AI look for patterns in the tags?
Yes. They basically asked the algorithm to find a pattern of methylation that correlated perfectly with chronological age, and it worked brilliantly.
Really?
Yeah, the algorithm found a specific set of a few hundred CpG sites, those cytosine-guanine pairings we discussed, where the methylation patterns changed with absolute predictable consistency as a human being aged.
So they just mapped it perfectly to time.
Right. These first-generation clocks were groundbreaking because they proved that we could take a simple blood sample, look at the epigenome, and accurately guess someone's chronological age. It firmly established the baseline metric for biological aging.
Right, but I imagine there is a major limitation there.
Oh, definitely.
Like, if I take a test and the odometer tells me I have the DNA methylation pattern of an average 45-year-old, that doesn't actually tell me much about my health risks, does it? It just tells me I look like an average 45-year-old on a molecular level.
That is the exact limitation. First-generation clocks were trained on chronological age, so by definition, they were just trying to guess your birth certificate.
Right.
They were not designed to predict disease. And that limitation led directly to the development of second-generation clocks, which are often referred to as morbidity or mortality clocks.
Morbidity clocks, wow.
Yeah, intense name. Instead of training the machine learning models on calendar years, researchers trained the algorithms on massive amounts of clinical health data.
What kind of clinical data?
They incorporated an individual's history of smoking, measured in pack years. They fed the algorithm data on levels of specific plasma proteins in the blood that are tightly linked to inflammation, kidney function, and liver health.
Okay, real physiological markers.
Exactly. They took all this physiological data and asked the AI to find the epigenetic signatures that predicted when a person would develop a chronic disease, or, frankly, when they were likely to die.
Wow.
These clocks do not measure calendar time. They measure your physiological risk of mortality.
I have to push back on this from a human perspective, though.
Go for it.
Because if I, as a listener, take a blood test and get a high score on a mortality clock, isn't that just a self-fulfilling prophecy of stress?
How do you mean?
Like if a piece of paper tells me my DNA says I am going to die 5 years sooner than expected, the sheer psychological anxiety of that revelation is going to flood my body with stress hormones, which, as we are going to discuss later, literally accelerates the aging process. It feels fatalistic.
That is a very common and honestly a very valid concern. And it raises an important question about how we use these powerful diagnostic tools clinically.
Right.
It is crucial to reframe what that score actually represents. A mortality clock is strictly a baseline measurement of your current trajectory.
Current trajectory, okay.
It's not a predetermined death sentence written in stone. Because the epigenome is plastic and modifiable, a high score on a second-generation clock is actually an actionable warning.
Oh. It is the biological check engine light.
Exactly. It tells you that the current state of your software is driving you toward disease. But because it is software, you can patch it.
You can change the code.
Right. If you receive a poor score and you use that information to fundamentally change your lifestyle, improve your diet, or utilize new therapies, you can retest a year later and physically watch that mortality risk decrease.
A clock score can go down.
Yes. It is a tool of empowerment, allowing you to track modifications rather than a tool of fatalism.
That makes a lot of sense. It provides a highly sophisticated biological feedback loop.
Exactly.
But looking at the sources, it seems even these mortality clocks had limitations, leading to a massive breakthrough with third-generation clocks.
Yes. The third-generation clocks represent a massive conceptual shift in how we track aging.
How so?
If the first generation was an odometer, this new generation is a speedometer.
A speedometer.
It does not give you a static biological age, like telling you, you are biologically 48. Instead, it measures the velocity of your biological decline. It tells you how fast you're currently aging.
A speedometer for aging. How do you even train an algorithm to measure speed rather than distance?
It required incredibly unique data. Researchers utilized a longitudinal birth cohort, study tracking over 1,000 individuals born in the same town in the same year, from birth all the way into midlife.
Oh, wow. So following them their whole lives.
Exactly. Over decades, researchers constantly tracked 19 different biomarkers of organ system integrity in these specific individuals. They tracked their lung capacity, their kidney clearance rates, their white blood cell counts, even the rate of their periodontal dental decay.
So they weren't just looking at a snapshot of a 50-year-old. They were watching the rate at which that specific 50-year-old's organs had degraded over the previous 30 years?
Precisely. They watched the organs decline, and then they found the specific epigenetic methylation patterns in the blood that correlated with the rate of that decline.
Okay, so the output is different.
Right. So the output of a third-generation clock is a pace. A score of 1.0 means you are aging exactly one biological year for every one chronological year. You are moving at normal speed.
Okay, 1-to-1.
But a score of 1.2 means your software is degrading 20% faster than average. For every calendar year that passes, your body absorbs 1.2 years of physiological damage.
Why is the speedometer approach considered such a radical breakthrough for anti-aging research compared to the mortality clocks?
It comes down to sensitivity and clinical utility. First and second generation clocks are heavily influenced by the accumulated damage of your entire life.
Right.
If you lived on fast food and smoked cigarettes for 20 years, that massive history of damage is baked into the odometer.
It's not going away overnight.
Exactly. If you suddenly wake up today, start eating a perfect diet, and exercise daily, it might take years for a second-generation clock to register a meaningful, statistically significant drop in your overall mortality risk. The sheer weight of your past masks your current progress.
But a speedometer doesn't care where you drove yesterday. It only cares how fast you are pressing the gas pedal right now.
Exactly. The sources indicate that third-generation clocks are sensitive enough to register changes in your pace of aging in just 3 to 6 months.
That's incredibly fast.
This is revolutionary for clinical trials and for individuals testing interventions. It allows researchers to administer an anti-aging drug or put a patient on a specific diet and see if the intervention is actually hitting the biological brakes in near real time.
We no longer have to wait 20 years to see if an anti-aging drug actually prevents heart attacks.
No, we can just look at the speedometer after a few months and see if the pace of decline has slowed.
Okay, so the diagnostic picture is clear. We now have the tools to precisely measure the speed at which our epigenetic software is degrading.
We do.
That naturally leads to the most exciting question in biology right now. What are the most advanced tools scientists are using to actually reach in and hit the brakes?
This is where it gets fun.
Yeah, we're moving from reading the buggy software to actively rewriting the code. And this leads us to the absolute frontier of longevity science, cellular reprogramming.
Cellular reprogramming is, without hyperbole, the most profound area of modern biology. It is based on a Nobel Prize-winning discovery that fundamentally rewrote the rules of cellular biology.
Let's hear it.
Researchers discovered that you can take a fully adult specialized cell, let's say a skin cell scraped off your arm, a cell that is fully aged and structurally committed to being skin,
Okay.
and you can expose it to a specific cocktail of 4 transcription factors.
And transcription factors are essentially master control proteins that turn large groups of genes on or off, right?
Yes, exactly. When the aged skin cell is exposed to these 4 specific factors, they dive into the nucleus and systematically wipe away the age-associated epigenetic marks.
Just erase them.
Yes, they actively strip off the methyl tags and reset the histone spools. As this happens, the cell physically forgets that it is old.
Wow.
But even more radically, it forgets that it is a skin cell. It reverts all the way back up the developmental timeline to become a pluripotent stem cell, a pristine, youthful, blank slate capable of dividing and transforming into any tissue type in the human body.
But wait, if we wipe its epigenetic memory completely, that sounds incredibly dangerous in the context of a living human being.
Oh, it is.
Right, because if I want to use this technology to rejuvenate my aging heart, I still absolutely need it to function as a heart. I don't want my left ventricle turning into a disorganized mass of blank slate stem cells that don't know how to contract and pump blood.
You have zeroed in on the exact danger that the field is grappling with. Fully reprogramming cells inside a living, breathing animal invariably leads to the formation of teratomas.
Teratomas, what are those?
A teratoma is a massive, chaotic tumor formed by cells that have lost their functional identity and just start growing wildly. You end up with a tumor containing random bits of bone, muscle, and hair tissue.
That sounds horrific.
It is fatal. This is why the entire frontier of the field has shifted focus to what is called transient or partial reprogramming.
How does partial reprogramming avoid the tumor problem?
The goal is an incredibly delicate, precise balance of time. Instead of leaving the reprogramming factors turned on for weeks until the cell becomes a blank stem cell, scientists administer the factors for just a short, highly controlled burst.
Like how short?
Perhaps just a few days. The aim is to push the cell back in time just far enough to wipe away the superficial age-associated epigenetic noise, to loosen those glitchy spools, but to actively stop the reprogramming process before the cell loses its underlying identity.
Okay, I'm picturing a smartphone again. It sounds like we are trying to trigger a factory reset to speed up the phone, clear the bloated cache, and get rid of the buggy apps draining the battery.
Right.
But we are desperately trying to abort the reset process right before it deletes the core operating system and turns the phone into a useless brick.
That is exactly it.
But mechanically, how does the cell remember what it is supposed to be if we are actively wiping its epigenetic memory?
That is the million-dollar question, and the current research suggests a fascinating answer. It appears that the epigenome is not just a single, uniform layer of tags. It has different layers of stability.
Layers of stability.
The epigenetic marks that dictate fundamental cellular identity. the core operating system that makes a heart cell, are deeply entrenched, highly stable, and resistant to change.
They're locked in.
Exactly.
Mhm.
However, the epigenetic marks that accumulate with age, the transcriptional noise and the buggy apps, are much more superficial and loosely attached.
So the reprogramming factors act like a chemical pressure washer.
Oh, I love that.
Right. Like a quick blast cleans off the superficial dirt of aging, but if you leave the washer on too long, it strips the paint down to the bare metal.
That's an excellent way to visualize it. By exposing the cell to the factors only transiently, the superficial aging marks are erased first, while the deep, identity-defining marks remain perfectly intact.
That's brilliant.
It is. Once the reprogramming factors are removed, the cell's internal machinery looks at those surviving core identity marks and uses them to reestablish a youthful, healthy epigenetic profile.
So it wakes back up.
Right. The cell wakes up, realizes it is still a heart cell, but suddenly it operates with the metabolic efficiency of a heart cell from 20 years ago.
And the preclinical successes detailed in the sources are almost unbelievable. We are talking about genuine age reversal in animal models.
Truly unprecedented outcomes. The sources detail extensive animal studies where cyclic partial reprogramming, meaning they genetically engineered mice so the researchers could turn the reprogramming factors on and off in cycles by basically feeding them an antibiotic.
They could just toggle it.
Yes.
Yeah.
And it actually extended the lifespan of mice suffering from accelerated aging syndrome
Extended their lifespan.
and health span. The mice looked physically younger, their spines straightened out, their organs functioned better. Furthermore, other studies have used this technique to successfully rejuvenate damaged heart tissue after a simulated heart attack, dramatically improving the tissue's ability to heal.
That's huge.
But perhaps the most stunning example in the literature was the restoration of vision.
Right, the optic nerve crush study. Let's really dig into the mechanics of that because it sounds like science fiction.
It really does. So in adult mammals, the central nervous system does not naturally regenerate. If an adult mouse or a human suffers severe damage to the optic nerve, the retinal ganglion cells at the back of the eye cannot regrow their connective axons to the brain.
The connection is just severed.
Right, and the resulting blindness is permanent. So researchers took aged mice, crushed their optic nerves, and then used a viral vector to deliver a partial subset of the reprogramming factors directly into the cells of the retina.
They delivered the software patch directly to the back of the eye.
Yes. And they only used 3 of the 4 factors, specifically leaving out the one most associated with cancer risk.
Smart.
Very. The transient reprogramming successfully reset the epigenetic state of those damaged retinal cells. It stripped away the silencing marks that prevented growth.
And what happened?
Astonishingly, those adult neurons regenerated their axons, threaded them back down the optic nerve into the brain, and actively restored visual function in the mice.
I could see again.
Yes, it proved definitively that aging and damaged cells retain a recoverable, youthful epigenetic blueprint deep inside them that can be coaxed back into existence.
Okay, so we can cure blindness and extend lifespan in mice. We have the biological command prompt. But mice are not humans. What is the catch here? Why isn't a partial reprogramming injection available in every pharmacy tomorrow?
The translational challenge moving from mice to humans is immense. The primary overriding hurdle is delivery and safety.
Getting it into the body safely.
Exactly. How do you deliver these incredibly powerful reprogramming genes into billions of specific cells scattered throughout a human body safely? You cannot just engineer a human embryo to have these genes turned on by an antibiotic like they did with the mice.
Obviously not.
You have to deliver them to an adult. And if your delivery vehicle is sloppy and leaves the reprogramming factors turned on for even a few days too long in just a tiny handful of cells in the liver or the lung, you risk initiating a fatal cancer.
The margin for error is essentially zero.
Exactly. This is why the first human clinical trials being planned are entirely focused on localized immune-privileged areas of the body, like the eye.
Oh, because it's contained.
Right. By injecting the gene therapy directly into the retina to treat conditions like glaucoma or macular degeneration, researchers can contain the reprogramming factors to a very small, isolated, enclosed area.
Which makes it safer to test.
Exactly. This minimizes any systemic risk to the rest of the body, while allowing them to prove the concept of epigenetic age reversal in living human tissue.
I see the bottleneck. Broad cellular reprogramming, even when we attempt to swing it carefully with transient pulses, is a biological sledgehammer.
It's a massive, blunt force intervention affecting the whole cell.
Right. So is there a way to be more precise? a way to put down the sledgehammer and pick up a scalpel, can we target specific aging genes without resetting the entire cell? Because that transitions us to the incredible use of CRISPR technology for epigenetic rejuvenation.
This is where the field becomes highly surgical. While transient reprogramming resets the entire cellular environment, CRISPR allows us to target a single, specific line of code.
Okay, let's break that down.
Now, most people have heard of traditional CRISPR-Cas9. It's famous for acting like molecular scissors.
Right, cutting the DNA.
You design a guide RNA to find a specific sequence of DNA, a specific gene, and the Cas9 enzyme physically cuts both strands of the DNA hardware. The cell tries to repair the cut, makes a mistake, and the gene is disabled.
It's a brilliant tool for treating certain genetic diseases.
It is. But cutting the genome is a permanent hardware alteration, and it carries the risk of off-target mutations, cutting the wrong piece of DNA.
And if we are trying to treat these subtle software degradation of aging, we don't want to go in and start permanently cutting the physical hardware.
Exactly.
So how do we use CRISPR for the epigenome?
We use a brilliantly modified version of the system called dead Cas9, or dCas9.
Dead Cas9.
Through precise protein engineering, researchers have gone in and deactivated the molecular nuclease domains of the Cas9 protein.
They broke the scissors?
They broke the scissors. The dCas9 protein can no longer cut DNA strands. However, it still retains its incredible, programmable ability to use a guide RNA to navigate through the billions of base pairs and bind to one highly specific, predetermined DNA sequence.
So if it can't cut the DNA, it just parks there. It essentially becomes a microscopic, highly precise delivery truck.
Exactly. It parks on the exact gene you want to target. And the genius is in what researchers attach to that delivery truck.
What are they attaching?
They can fuse dCas9 to the specific epigenetic enzymes that naturally add or remove the methyl tags we discussed earlier.
Oh, wow.
For example, there's an enzyme called TET1, which functions to oxidize and eventually strip away methyl tags. If you fuse dCas9 to TT1, you create a microscopic machine that can navigate to a specifically silenced gene, park on the promoter region, and actively strip away the methyl tags.
Turning that specific gene back on.
Turning it back on.
And I assume you can do the reverse.
Yes. If you have a pro-inflammatory gene that is causing inflammaging because its spool got too loose, you can fuse dCas9 to an enzyme called DNMT3A.
And that adds the tags.
Yes. The truck parks on the inflammatory gene and actively adds methyl tags, dimming the switch and silencing the inflammation.
We are literally building custom programmable dimmer switches. Like, we can manually go into the nucleus, find the exact line of code that is glitching, and turn up the volume on a repair gene or muted gene causing damage.
It is the ultimate software patch. And the preclinical applications detailed in the sources are fascinating.
Like what?
Researchers have used this dCas9 system in adipose, or fat tissue, to edit specific methylation patterns. By targeted switching of certain metabolic genes on and off, they are attempting to fundamentally remodel the tissue to combat obesity and metabolic dysfunction at the epigenetic root, rather than just treating the symptoms with weight loss drugs.
That's incredible. But I want to zero in on one specific application from the sources that sounds incredibly profound: telomere rejuvenation.
Ah, yes.
Because we constantly hear about telomeres in the longevity space.
Yeah.
They are the protective caps at the ends of our chromosomes, often compared to the plastic tips on shoelaces.
Great analogy.
And every time a cell divides, the copying machinery cannot quite reach the very end of the DNA strand, so the telomere gets a little bit shorter.
Right.
And when they burn down too far, the cell senses the damage, hits senescence, and becomes one of those toxic zombie cells.
Exactly. Telomere attrition is a major fundamental limit on cellular replicative lifespan. Now, biology does have a built-in solution.
It does.
There is an enzyme called telomerase that can actively rebuild and lengthen these caps. But the gene that produces telomerase, the TERT gene, is heavily methylated and permanently turned off in almost all of our adult somatic cells.
Wait, if the body has the blueprint to rebuild the caps and prevent cellular aging, why does it aggressively keep that gene locked away?
It's a critical evolutionary defense mechanism against cancer.
Oh.
Yeah. If every cell in your body could endlessly rebuild its telomeres, any cell that mutated could divide infinitely, rapidly forming a tumor. By keeping the TERT gene silenced, the body puts a strict limit on how many times a mutated cell can divide before it naturally burns out and dies.
But the sources show researchers are using this dead CRISPR delivery truck to go in and turn the TERT gene back on. Isn't that asking for cancer?
It would be, if done systemically. But they're utilizing this technology in a highly controlled, highly specific context. Primary resting T cells.
T cells, the immune system.
Right. T cells are the foot soldiers of your immune system. As we age, our T cells are forced to divide repeatedly to fight off lifelong infections, like cytomegalovirus. Over decades, their telomeres get critically short, the cells become exhausted, and our immune system fundamentally weakens.
Which is why older people get sick more easily.
Exactly. This immune exhaustion is a major reason why older individuals are so vulnerable to novel viruses and why vaccines are less effective in the elderly.
So how are they fixing the T cells?
Researchers are extracting these T cells from the blood. Then, using the DCAS9 system fused with activating enzymes, they target the TERT gene promoter ex vivo.
Ex vivo meaning they perform the gene editing outside the body in a controlled laboratory environment.
Exactly. By locally removing the repressive methyl marks specifically on that one gene, they successfully reactivated telomerase expression in the T cells.
They turned the telomerase factory back on. What happened to the cells when they were tested?
The activation significantly extended their replicative lifespan. The T cells could divide far more times while maintaining a youthful, vigorous immune response.
So they were rejuvenated.
Yes. The telomerase successfully rebuilt the protective caps on their chromosomes. And crucially, because it was an epigenetic edit, a software patch, and not a permanent hardware change, the activation was transient.
So it didn't stay on forever.
No. The methyl tags slowly returned over time, turning the gene back off before the cells could cross the line into uncontrolled tumor-forming growth.
Man, it is brilliant, but I have to play the skeptic here again.
Please do.
Extracting T cells, editing them in a billion-dollar clean room, and infusing them back into a patient is one thing. We already do that for certain cancer therapies, and it costs hundreds of thousands of dollars. But if aging is a systemic problem affecting the heart, the brain, and the liver, how do you get these microscopic CRISPR delivery trucks into millions of solid tissue cells inside a living, breathing human? Won't the human immune system just recognize the CRISPR proteins as foreign invaders and destroy them before they even reach the target DNA?
You have identified the single most significant bottleneck in all of gene therapy and systemic epigenetic editing right now. Delivering these large CRISPR-Cas9 complexes safely in vivo inside the living body is incredibly difficult.
Because the body fights back.
Exactly. We have viral vectors, like adeno-associated viruses, or AAVs, which are highly efficient at infecting cells and delivering genetic payloads. But AAVs can trigger massive, sometimes fatal immune responses.
Oh, so that's out.
Well, then we have non-viral methods, like the lipid nanoparticles that were made famous by the mRNA COVID vaccines. They are much safer. But currently, when you inject lipid nanoparticles into the bloodstream, they almost all get swallowed up by the liver.
So they don't even make it.
Right. They struggle to efficiently reach the heart, the brain, or the muscles.
So the delivery trucks are getting stuck at the first toll booth.
Exactly. The field is aggressively working on developing localized delivery systems or engineering tissue-specific promoters that ensure the DCAS9 is only active exactly where we want it. But until we solve this systemic delivery problem, localized surgical gene editing for anti-aging remains highly experimental in humans.
Which begs an immediate practical question.
What's that?
If partial cellular reprogramming risks teratomas and CRISPR-Cas9 is still largely stuck in the lab struggling with delivery vehicles, what tools do we actually have right now? What pharmacological compounds are currently being tested in human clinical trials to shift this epigenetic landscape systemically today?
This is where the science moves from the theoretical realm of mice and petri dishes into clinically observable human data. And the most provocative, widely discussed example in the sources is a landmark clinical trial focused on immune and thymus regeneration.
Okay, let's lay the groundwork for this. What is the thymus, and why is it so critical to aging?
The thymus is a small gland located in your upper chest, just behind your sternum. It serves as the primary training ground for your immune system, specifically for the maturation of T cells.
The soldiers.
Right. However, the thymus undergoes a biological process called involution. Starting right around puberty, the active lymphoid tissue of the thymus begins to shrink and is progressively replaced by useless fat cells.
Just turns to fat.
Yeah. By the time a human being is 60 years old, the gland is mostly fatty, non-functional tissue. This involution is a primary driver of the immune exhaustion we discussed earlier. You simply stop producing fresh, naive T cells capable of fighting off new threats.
So if the thymus is essentially just a lump of fat by the time you're 60, how on earth do researchers convince that fat to turn back into functional, active immune tissue?
They utilized a highly specific synergistic protocol combining three existing well understood FDA-approved drugs.
Okay, what were they?
Recombinant human growth hormone, DHEA, which is a mild over-the-counter steroid hormone, and metformin, which is one of the most common diabetes drugs in the world.
Why those three specific drugs? What is the mechanism there?
The growth hormone was the primary biological driver. Previous animal studies had shown that administering growth hormone can stimulate the proliferation of thymic epithelial cells, essentially coaxing the gland to rebuild its functional architecture.
So the growth hormone tells it to grow.
Exactly. However, growth hormone has a severe known side effect. It rapidly induces insulin resistance. It causes the liver to pump out more glucose while preventing muscles from absorbing it, essentially pushing the patient toward type 2 diabetes.
You definitely don't want to trade an aging immune system for diabetes.
Precisely. Therefore, the researchers had to counter that side effect. That is why the DHEA and metformin were included in the protocol. Metformin activates a metabolic pathway called AMPK, which forcefully pulls glucose out of the blood and into the cells, restoring insulin sensitivity.
So it's a balancing act.
Very much so. The three drugs were designed to work in synergy. The growth hormone rebuilds the thymus, while the metformin and DHEA act as a biochemical shield against the side effects. They administered this cocktail to a cohort of healthy adult men over the course of 12 months.
And the outcomes of this trial were kind of historically staggering, weren't they?
They genuinely shocked the field. First, MRI imaging of the participants' chests showed actual physical regeneration of functional thymus tissue, replacing the fat.
It actually regrew.
It did. But the real breakthrough was when they tracked the participants using those epigenetic mortality clocks we discussed earlier.
The software checkers.
Right. On average, the participants did not just slow down their aging. They reversed their biological age by 2.5 years compared to where they started the trial.
According to the software check, they went backwards in time.
Yes. And what is even more fascinating is the rate of that reversal. The data showed a nonlinear acceleration.
What does that mean?
During the first 9 months of the 12-month trial, biological age reversed at a rate of about 1.6 years per year. But in the final 3 months of the trial, the rate of reversal accelerated massively to 6.5 years per year.
Whoa, it sped up.
Yes, this strongly suggests that the epigenetic remodeling effects compound over time as the immune system comes back online. Even more encouraging for the researchers, the biological age reduction persisted even 6 months after the trial ended, and the men stopped taking the drugs.
Okay, this sounds like a miracle cure, but I am looking at the data from the sources, and I see a massive paradox that we have to address.
Okay, let's hear it.
In that exact same thymus trial, when the researchers measured the participants' physical fitness, specifically their VO2 max, which is the absolute gold standard for measuring cardiovascular health and endurance, it stayed completely flat. It didn't improve by a single point. So if the epigenetic clock says your cells are 2.5 years younger, but the patient doesn't feel any fitter and their heart and lungs aren't pumping oxygen any better, what is this clock actually measuring? Is biological age just a vanity metric on a piece of paper?
That is an incredibly astute observation, and it highlights exactly what these clocks are and are not measuring. We have to remember how second-generation clocks are built. They are trained to predict the onset of disease and mortality risk, primarily based on cellular decay, inflammation, and immune function in the blood. They are highly sensitive to shifts in systemic inflammation.
Right.
The immune system regeneration driven by the thymus trial fundamentally lowered the participants' future risk of developing age-related diseases like cancer or severe infections. The clock accurately captured that profound shift in underlying biological risk.
But lowering your risk of cancer doesn't automatically give you bigger muscles or better lungs.
Correct. Reversing epigenetic age in the immune system does not magically rebuild atrophied muscle fibers or clear calcified plaque out of a coronary artery instantly.
Ah, I see.
Biological aging is highly multidimensional. Different organ systems age at different rates. A specific peptide protocol might powerfully reboot the immune system software, which the blood-based clock eagerly detects, without inherently changing the physical architecture of the cardiovascular system.
It's just one piece of the puzzle.
Exactly. It proves the pharmacological intervention works at a molecular level, but it also proves that true human longevity will require a multimodal approach. We can't just fix the blood. We have to fix the heart, the brain, and the muscles.
Speaking of molecular interventions, the sources also dive deep into smaller, more targeted pharmacological tools that are widely available, specifically focusing on NAD plus precursors.
Yes, small molecules are a massive area of intense clinical focus. NAD plus nicotinamide adenine dinucleotide is a critical coenzyme found in every single living cell.
Every cell.
Every single one. It is absolutely essential for the electron transport chain in your mitochondria. Without it, you cannot turn food into energy and you would die in seconds.
So it's pretty important.
Very. But crucially for longevity, NAD+ is also the required fuel source for a highly specific class of epigenetic maintenance enzymes called sirtuins.
What do sirtuins do?
Sirtuins are heavily involved in maintaining those tight histone spools we discussed at the very beginning. They act to repair DNA damage and ensure the spools stay tightly wound, silencing the noise.
So they're the maintenance crew.
Exactly. But here's the problem. Sirtuins are NAD dependent. They cannot function without consuming NAD plus Rs. And as we age, our natural cellular pools of NAD plus plummet drastically. The enzymes are sitting there ready to maintain the software, but they're completely out of gas.
So the epigenetic software glitches faster simply because the cellular mechanics don't have the fuel to do their job.
Exactly. This is why supplementing with NAD plus precursors like NMN or NR has become such a prominent strategy.
Right, you hear about NMN all the time.
Because these small molecules are easily absorbed by the cell and then converted directly into NAD plus pool. By artificially boosting the intracellular NAD plus pools, you are essentially refueling those sirtuin repair enzymes, allowing them to resume their job of maintaining youthful epigenetic patterns and tightly wound DNA spools.
That makes total sense.
Furthermore, researchers are exploring the powerful synergy of combining different classes of these epigenetic drugs.
Synergistic in what way?
Well, for example, combining a drug that removes methyl tags with an HDAC inhibitor. An HDAC inhibitor is a drug that prevents the cell from removing those acetyl groups from the histone spools, thereby keeping the chromatin physically open and accessible.
Oh, right, the open vault.
Exactly. By combining them, researchers can powerfully and forcefully open chromatin and restore gene expression in heavily aged cells. In animal models, this dual pharmacological approach has shown significant rapid improvements in cognitive performance by turning neuroplasticity genes back on in the brain.
Which brings us to the final, and realistically the most important part of this entire deep dive.
Let's do it.
Because we have spent the last hour talking about absolute sci-fi technology, transient cellular reprogramming, viral vectors delivering dead CRISPR trucks to the retina, custom hormone peptide stacks to regrow organs.
It's heavy stuff.
It is. But for the listeners sitting in traffic right now, or cooking dinner, experimental CRISPR therapies are completely out of reach. Yet the sources make one thing incredibly clear. The most validated, most universally accessible, and perhaps most powerful epigenetic interventions on the planet right now are your daily behaviors.
This is perhaps the most empowering realization in all of modern biology. We must fundamentally stop framing lifestyle choices, like what you eat, how you move, and how much you sleep, as just generic, colloquial health advice.
Right, it's not just eat your veggies.
Exactly. Scientifically speaking, at a molecular level, these behaviors are active, precision epigenetic modulators. They are the most powerful levers you currently possess to actively manipulate your cellular software.
So let's break down the mechanics of these daily habits, starting with diet and fasting. We know eating vegetables is good for us, but what is a vegetable actually doing to the DNA?
The way you eat directly provides the chemical substrates needed for epigenetic regulation. Let's look at caloric restriction first.
Okay.
The sources detail a rigorous, landmark 2-year randomized controlled trial where healthy adults simply reduced their overall caloric intake by 25%. They didn't change what they ate, just how much.
And what happened?
Using the advanced third-generation speedometer clocks, researchers found that this basic caloric restriction actively slowed the pace of biological aging by 2 to 3%.
Wait, why does simply eating less slow down the degradation of the software?
Because chronic overnutrition, constantly flooding the body with glucose and amino acids, hyperactivates nutrient-sensing pathways in the cell, most notably a pathway called MTO.
mTOR.
Right, when MTOR is active, it tells the cell to grow, build, and divide, which naturally accelerates epigenetic drift and cellular exhaustion.
So it's constantly pushing the cell to work.
Yes. Fasting, or caloric restriction, deprives the cell of nutrients, which flips a metabolic switch. It turns off the mTOR growth pathway and turns on an energy-sensing pathway called AMPK.
Which we mentioned earlier.
Yes. Activating AMPK triggers cellular repair, boosts those critical NAD+ levels, and activates the sirtuin repair enzymes. It literally forces the cell to stop dividing and start cleaning house.
But it isn't just how much you eat. The sources talk specifically about what you eat changing the epigenome.
Yes, because the enzymes that place the methyl tags on your DNA do not create those tags out of thin air. They have to extract them from your food.
Oh,
Another clinical trial utilized an 8-week structured program combining a specific diet, exercise, and relaxation. The diet was heavily focused on what researchers call methylation-supportive foods: dark leafy greens, cruciferous vegetables like broccoli, pumpkin seeds, and liver.
what makes those foods methylation supportive?
They are incredibly rich in specific micronutrients like folate, vitamin B12, betaine, and choline. These molecules feed directly into the body's methionine cycle to produce a compound called SAMe.
Same?
Right. SAMe is the universal methyl donor for the entire body. It is the literal physical building block that your enzymes use to create those methyl tags.
So if you don't eat those foods,
If your diet is deficient in these greens and nutrients, your body physically cannot manufacture the tags needed to maintain the epigenetic software. Your dimmer switches fail because they lack raw materials.
wow.
That specific 8-week dietary program resulted in an astounding 3.23-year reduction in biological age. Similarly, long-term adherence to a polyphenol-rich Mediterranean diet has been shown to consistently attenuate the biological clocks.
Okay, let's talk about exercise. I understand how running on a treadmill strengthens the heart muscle, but what is a bicep curl or a jog doing to the DNA hidden deep inside the nucleus?
The clinical data shows that engaging in 150 to 300 minutes of moderate aerobic exercise per week, combined with resistant strength training, actively lowered the pace of aging by 3 to 7%.
Just by moving.
And it achieves this through a highly direct mechanical and chemical signaling pathway. When you physically contract your skeletal muscles, you burn ATP for energy. This alters the ratio of metabolites inside the muscle cell.
Okay.
The cell senses this energetic stress and releases signaling molecules, including specific interleukins and AMPK.
So the physical tension creates a chemical distress signal.
Exactly. And that chemical signal travels directly into the nucleus and physically alters the DNA methylation patterns.
It reaches right into the DNA.
It does. The exercise specifically strips methyl tags off of genes related to energy metabolism and mitochondrial biogenesis, turning them on to build more energy factories, while simultaneously adding methyl tags to suppress pro-inflammatory genes. The physical act of exercise is literally a command prompt turning on the youth-associated software in your muscle tissue.
And then there is the silent killer that undoes all of this hard work: sleep and stress.
We cannot overstate the importance of the circadian rhythm, your body's internal 24-hour biological clock. This rhythm is deeply physically intertwined with the epigenome. The sources show that consistently getting less than 7 hours of sleep fundamentally disrupts the core clock genes in your brain and peripheral tissues. And here's the key. These clock genes are physically coupled to the epigenetic maintenance enzymes. The enzymes do their repair work on a strict schedule, mostly while you sleep. When your sleep is chaotic, the timing of that epigenetic maintenance is destroyed. The data shows rapid detectable acceleration of biological age clocks within just weeks of severe sleep disruption.
I am looking at the data on chronic psychological stress, and this is the mechanism that absolutely blows my mind.
It's fascinating.
Because I can visualize how a chemical from a broccoli floret or the physical contraction of a muscle fiber can alter cellular chemistry. But how does the abstract psychological burden of financial anxiety or a toxic boss physically reach into the microscopic nucleus of a cell and move a chemical methyl tag?
It is a profound mechanism, and it happens through a highly physical biological cascade known as allostatic load.
Allostatic load.
When your brain perceives a chronic psychological threat, say relentless anxiety about debt, your amygdala signals your hypothalamus, which triggers your pituitary gland, which sends an alarm down to your adrenal glands. Your adrenal glands respond by flooding your bloodstream with cortisol and adrenaline.
The fight or flight response?
Yes, but it's not just an emotion. It is a systemic flow of powerful steroid hormones. Cortisol enters your cells, binds to specific glucocorticoid receptors in the cytoplasm, and that newly formed receptor complex transits directly through the nuclear membrane and into the nucleus where the DNA is stored.
Wait, it goes right to the DNA.
Yes. Once inside, the cortisol complex acts as a massive transcription factor. It physically sits on top of the DNA strand. It actively blocks the enzymes that normally maintain your youthful epigenetic marks and simultaneously recruits other enzymes that aggressively strip the methyl tags off of inflammatory genes, turning them on while silencing your immune repair genes.
The anxiety is literally physically bathing the DNA in an inflammatory signal.
Yes. Over months and years of chronic stress, this constant hormonal signaling forces the epigenetic enzymes to permanently alter the methylation patterns to favor this high-alert, inflammatory, degrading state.
That's terrifying.
Your psychological stress physically rewrites the software to age you faster. This is exactly why chronic stress visibly accelerates the epigenetic clock.
And there's a flip side, right?
Yes. Conversely, the sources highlight that interventions like mindfulness meditation, structured breathwork, and time spent in nature directly reduce this allostatic load. By lowering the systemic cortisol signaling, you remove the heavy physical pressure on the epigenome, allowing the sirtuins and maintenance enzymes to restore normal, useful gene expression. It is a direct, measurable, mechanical pathway from your mental state to your DNA.
It really all comes back to the software.
It does.
So as we wrap up this deep dive, let's summarize the incredible journey we have taken today through these sources. We started by entirely dismantling the old dogma that aging is just inevitable thermodynamic resting.
Right, throwing that old idea out.
We learned that the epigenome is a dynamic, highly programmable layer of software controlling our DNA hardware. We saw how third-generation clocks act as real-time speedometers, allowing us to measure the exact velocity of our biological decline.
Which is a huge leap forward.
We explore the absolute sci-fi frontiers of hitting the factory reset button with transient partial cellular reprogramming and the surgical precision of using dead CRISPR delivery trucks to act as custom dimmer switches on specific aging genes.
Those programmable patches.
We looked at how targeted pharmacological compounds can reboot the immune system, and finally, we grounded it all in the undeniable mechanistic reality that our daily lifestyle choices, what we eat, how we move our muscles, and how we manage the cortisol of stress, are actively rewriting our epigenetic code every single day.
It is a remarkable, comprehensive synthesis of where the science stands today. And I want to leave you, the listener, with a final provocative thought to ponder as you go about your day.
Please do.
We have established that third-generation clocks can measure the pace of aging in real time. We have seen that the field is on the absolute cusp of safely utilizing transient cellular reprogramming and epigenetic editing in living humans.
Yeah.
If these current scientific trajectories hold, we are rapidly approaching a profound societal threshold. We might soon transition away from a reactive medical paradigm, a society that waits for catastrophic diseases like Alzheimer's, cancer, and heart failure to occur before desperately trying to treat the physical symptoms.
Instead, we get ahead of it.
Exactly. Instead, we are moving toward a proactive paradigm where we routinely update our biological software, stripping away the epigenetic noise to prevent those diseases from ever manifesting in the first place.
Wow.
This raises an extraordinary question for all of us. What does human life, society, and purpose actually look like when we begin to view aging, not as a fixed, inevitable timeline leading to decay, but simply as a highly treatable condition?
A treatable condition. That framing, it changes absolutely everything. It means the symphony conductor doesn't have to keep playing chaotic noise until the orchestra collapses. We might just be able to print him some new sheet music. Thank you for taking this deep dive with us.